care homes.
And maybe get a beverage too. If you have a chance to go to that.
Yeah, do that. 10 years ago, nobody wants it. We're gonna take a bath on it.
I'm not on the highway.
So it's actually a gamble.
You need a drink? Go grab a drink too.
Ultimately, I think the consensus is
You
that
need-
it's gonna last. Pushing for it. To some extent.
Hello, everyone. We'd like to go ahead and get started. We'd like to thank you for being here today. Those of you that are joining us in person and those of you that are joining us remotely, again, thank you. I'm standing here with Karen Schmitz. We are the nurse practitioners, part of the Brain Tumor Institute. We also have Chris Balmacetta is at the back of the room, and Laura Jaros, who is running around with her computer, and Eliza Delgado, who was registering you, and Meg Schwartz, who is away on vacation, but we're all your program coordinators for today's event. But we'd also like to thank our program directors, including Dr. Stoop and Dr. Chandler, for being part of the leadership of the MBTI. We do have a little bit of some housekeeping items. Masks are not required in this room. It's to your own personal discretion. But if you do leave the room, you need to have your mask on. Important information is the restrooms are outside the door and around the corner near L North. We will be having today's session. We will have 2 speakers, then a dinner. Then we will have 1 more speaker, and then we will break for our support group. Questions for today's, if you are in person, there are notebooks, notepads at your place settings. Please write down a question, and you can either get it to Karen, myself, or Chris. Chris will be fielding them. He will also be fielding them on the chat room. So if you have any of that, please fill out the questions and we'll try to get to them. We'd also like to give a special thanks to Vincent Brock from the American Brain Tumor Association. Please be sure to visit him at the table. He's provided some information at your place settings. Otherwise, those of you that are remote, you may also go online to take part in any of the information that the ABTA has to offer or future events that they may. Did I miss anything? Maybe hosting.
You're doing great.
Okay. Our first speaker is Dr. Craig Horbensky. He is Director of Neuropathology, Director of the Brain Tumor Bank. His clinical focus is on genetic testing of brain tumors, and specifically glioma and meningiomas. Today, he's going to be talking to us about seizures in glioma patients. Thank you, Dr. Horbensky. Oh, I didn't even.
Hello, everybody. Thanks very much for coming. And one of the projects that we work on in my laboratory, we have about a dozen going on, roughly speaking, thus far, including some work with Dr. McGill on meningiomas. But one of the projects that's just reached fruition to some extent after 5 years of working on it is our project on seizures in glioma patients. Now, as a background, it's a lot more common than a lot of people might realize, is that over half of patients with gliomas will have at least 1 seizure during the course of their disease. Very often they'll have multiple seizures, and that's called tumor-associated epilepsy. And where it's somewhat paradoxical, at least historically, that they would be more common in relatively younger patients and more common in lower grade tumors than in grade 4 glioblastomas. But there actually turns out to be a reason for that. But we knew that for many, many decades, that somewhat paradoxically lower, less aggressive tumors would be more likely to trigger a seizure. Now, the frontline therapy, meaning the first treatment that people typically get when they have a seizure, is Keppra, also known as levetiracetam. And that's fine. It works very well. It's pretty well tolerated. But at least in our study internally of patients here at Northwestern, the vast majority of them will have at least one breakthrough seizure, meaning they'll have a seizure while they are on Keppra. So the Keppra failed to prevent that seizure from happening. And that typically will then lead to adding on other anti-epileptic drugs, different drugs that have more power or more adverse side effects. And so it really becomes a serious quality of life issue. It's not something to be dismissed, especially for somebody who's just gone through a seizure. To make matters worse, there's emerging literature now that bursts of neuronal electrical activity can themselves actually encourage gliomas to progress. The electrical activity from the firing neurons actually has been shown to stimulate glioma cells to both proliferate and migrate further and faster through the brain. This is just research that's gotten a lot of attention in the brain tumor community over the last couple of years. So there's a lot of very good reasons to try and limit seizure activity in glioma patients. Now, what I'm about to show you next needs a little bit of introduction. First of all, the World Health Organization classification scheme for central nervous system tumors. This is our Bible of sorts that we use to codify all the different kinds of tumors and give them different names and everything. Well, back in 2000, that was my, does this laser pointer work? Okay, not too well. Back in 2000, that's how fat the WHO book was. You can see how it's gradually gotten heftier, and the one that just came out a year ago in October of 2021 broke the record at 568 pages. And the reason for that is over the last decade, 2 decades, there's been an explosion in the amount of molecular data, of genetic data on brain tumors, both in kids and in adults, that has necessitated us developing a more sophisticated classification system, because we're finding out that two tumors that we thought were the same under the microscope, one has mutation X, the other has mutation Y. And when you follow the patients over time, their clinical courses vary greatly. So that has required us to develop a more complicated and a more comprehensive, but a more accurate classification system. So this is the cover of that. And I say that as a shameless plug because this was the first edition where I was able to contribute as a co-author to one of the chapters. And as luck would have it, one of my photomicrographs made the cover. So anyway, there's some key words though to understand this WHO scheme. First of all, when something or someone does not have a particular mutation, whatever mutation you're talking about, we refer to them as wild type, meaning they're representative of what you would encounter in the wild, so to speak. Mutant, of course, does have the mutation. The genes in question here that we're going to be focusing on for the duration of this presentation are very interesting genes, isocitrate dehydrogenase 1 or isocitrate dehydrogenase 2. We combine them to call them just IDH for simplicity. And they're very interesting little enzymes, and we'll see why as we progress through this. But there's this idea of IDH wild type gliomas and IDH mutant gliomas. And so try to keep those in your mind. And the reason for that is that in the 2016 scheme for the World Health Organization, they were the first to start using these IDH wild type and IDH mutant terms in many of the official diagnostic lines. 2021, this was simplified to some extent. And now we call all those IDH wild type glioblastomas and IDH mutant astrocytomas and oligodendrogliomas, we call them adult type diffuse gliomas. And we use that term, the supercategory term, because we're referring, this is a tumor that the tumor genetics is typical of what you would encounter in an adult patient. So a 14-year-old kid, a very unfortunate 14-year-old kid, could get an IDH mutant astrocytoma. It is still considered an adult type diffuse glioma, even though, of course, that kid is not legally or really biologically an adult yet. And it is really important to keep that in mind, this 3-tiered schemes. You got IDH wild type glioblastomas, IDH mutant astrocytomas, and IDH mutant oligodendrogliomas. All adult type diffuse gliomas come in one of those 3 flavors. And it's important, and the reason for that is because when you track their outcomes over time, they do differ substantially in terms of outcomes. So the IDH mutant oligodendrogliomas, that's the blue line, they're the ones that by definition also have to have not just the IDH mutation, but one P19Q co-deletion. It's a specific chromosomal alteration that really distinguishes them from all the other gliomas. And those patients on average have the longest survival. Of course, individuals can vary greatly. IDH mutant astrocytomas lack that 1p19q codeletion. That's the gold line. And they have a survival that's a little bit worse on average than oligodendrogliomas, but still relative to IDH wild type glioblastomas, which is the red line, they actually do pretty well. Although ultimately, all three of these adult type diffuse gliomas are usually fatal in the majority of cases. Now that IDH mutation is interesting. Normally, this is a humble little enzyme, IDH1 and IDH2, and they convert something called isocitrate into alpha-ketoglutarate in the cell. Every med student and every doctor remembers biochemistry, both in undergraduate as well as in medical school, because we all had to memorize these biochemical pathways in the cells. And at the time when we're memorizing these pathways, we're trying to figure out why on earth we are being taught this stuff. How are we ever going to use it in our clinical practice? Well, guess what? We did. And because the IDH mutant enzymes, instead of making alpha-ketoglutarate, they make this aberrant chemical called 2-hydroxyglutarate or D2-hydroxyglutarate. It's a chemical, it's a mysterious little chemical that the cells normally don't make much of and really don't seem to want it around. But cancer cells, many cancer cells, many cancer types will produce a lot of this stuff. And so there's been a ton of research since this was discovered back in late 2009. There's been a ton of research worldwide as to what this chemical is doing to cause gliomas and how does this chemical change the biological behavior of those gliomas. Well, we've taken a slightly different tack on that. We're not studying so much directly what that 2-hydroxyglutarate is doing to the glioma itself. We're studying what it's doing to the brain around the glioma, because that stuff gets produced and released into the brain. And what we found originally, this is going back in 2017, 3 different cohorts totaling over 700 patients from 3 different hospitals. And we tracked in their symptoms at the time of clinical presentation. In other words, what caused them to go seek medical help when they were first diagnosed with a glioma? Was it a headache? Was it paralysis? Was it some sort of tingly feeling or whatever? It turns out that most IDH mutant glioma patients, these are patients whose tumors ultimately were proven to have IDH mutations. These are the white and blue bars. Most of them had seizures as part of their clinical presentation, whereas most glioblastoma patients did not. Those are the golden or yellow bars. And that's even though glioblastomas are more aggressive and more destructive than those IDH mutant tumors. Whether they're astrocytomas, which are the white bars, or oligodendrogliomas, which are the blue bars, didn't matter. Those tumors, even though they're less destructive, they caused a lot more seizures than the wild type tumors did. And that was interesting because, as it turns out, when you really look at the chemical structure of that weird little compound called D2-hydroxyglutarate, it's almost identical to another chemical that's very important for all of us, and that's glutamate. The only difference is this OH here versus this H2N over here. Glutamate is the most widespread excitatory neurotransmitter in the brain. In other words, that's the chemical that your brain makes when it wants your neurons to fire. So D2-hydroxyglutarate looks an awful lot like that glutamate, doesn't it? I mean, chemically, they're very, very similar. And with the help of an old friend of mine, Pam Line from UC Davis, we trained at the same neuroscience laboratory back at the University of Buffalo, which is my hometown. And she went on to UC Davis to become the queen of environmental neurotoxicology. She helped us in these early experiments culture neurons. These are rat neurons, rat brain neurons on these grids. And these little electrodes, they'd be sitting on these electrodes, and electrodes would just physically record the electrical activity. It'd be like sticking a couple of wires into a battery and getting voltage. Here's what happens when you treat those cultured cortical neurons with D2-hydroxyglutarate. So you can see on the right the massive hot bursts of electrical activity in these cultured cortical neurons. That's basically the cell culture equivalent of a seizure, what you're seeing on the right. What you're seeing on the left is just normal activity. So right off the bat, we have this chemical product of IDH mutant enzyme that seems to be really irritating the neurons. And it matches perfectly the clinical presentation of these patients, which is typically seizures. In fact, that actually made the cover of Neurology back in 2017. So that was the old work that we had done on the subject. The current work, which has been 5 years in the making, took that to the next level. And that's thanks mostly to Michael Drum, who's an MD PhD student in my laboratory. He's wrapping things up. He'll be defending his thesis next spring, and he's a superstar. Well, he tracked over 250 glioma patients here at Northwestern with the help of Dr. Jessica Templer, who is a neuro-oncologist specializing in tumor-associated seizures. So she was the perfect person to work with us on this. And what they both found was when patients were getting, so we already knew what patients were like at the time of original surgery, right? In other words, we already had good papers and good data on the symptoms they had before, they underwent surgery. But there wasn't nearly as much known about what happens once the patient's operated on. When they're operated on, they get radiation, they get chemotherapy, what happens to their seizures at that point? How do they change over time, and what factors affect whether somebody's going to get a seizure during their disease or not? And what we found was actually, we can actually, I'm going to skip a couple of just in terms of speed, because I know we start a little late and I want to be mindful of time. There's basically, with the help of Dr. Kirsten Bell-Burdette, who's a biostatistician who helped us with all this work, we found 4 things that really affect the risk of patients who are going to have seizures. Now, we were able to tell with a fairly high degree of accuracy who's going to develop a seizure after surgery and who's not. One is the genetic profile of the glioma. Does it have an IDH mutation or not? That's a big one. Where is that glioma located? Is it in the frontal lobe? If it is, the risk goes up. How much of that tumor was taken out at the time of surgery? Was it a lot? Did they pretty much get more or less the entire tumor out as visually? Or did they know they left some behind? Because if they got more out, not too surprising, seizure risk went down. The biggest one by far, though, is whether the patient had a seizure before surgery. In other words, did they have a preoperative seizure? Did they have a seizure as part of their clinical presentation? If they did, not too surprising, their risk of having additional seizures once they were operated on and once they got treatment, their risk was several-fold higher than other people. So what we're doing now is actually, we've already gotten that model to work really well in this original cohort of about 250 patients. We have another 350 patients that we're tracking over time now to validate this model. So what we're trying to do is develop a web-based app where you can punch in all of those variables. Anybody could do this. And then determine your risk of developing a postoperative seizure 1 month after surgery, 3 months after surgery, 6 months after surgery, a year after surgery. And this will be very useful, we hope, for patients and their families, their caregivers, as well as any treating physicians who are trying to manage their seizures and decide, okay, who needs to be on Keppra or other anti-epileptic drugs up front versus who can we get away with just watching them and spare them the hassle of having those medications. Oh, and by the way, Kirsten just had, there's a sidebar, she just had her little nugget. So she's on maternity leave. This is actually the last project she wrapped up before going on maternity leave. And there's that little guy right there. So one of the things that Michael did in my laboratory that was really insightful, he took those cultured cortical neurons. I showed you the neuronal cultures already. And what he did was he treated them with D2-hydroxyglutarate, like what we had done before, but he did it in a different way. He tried the neurons just by themselves. So there's just neurons sitting in the culture dish, or neurons that also have glial cells. These are not glioma cells. They're not cancerous cells. They're just normal astrocytes and oligodendrocytes sitting there with the neurons. And that's important because actually, even though you think of your brain as being packed with neurons, and it is, there's actually 10 times as many glial cells in your normal brain as there are neurons. And these glial cells are extremely important in helping the neurons survive and thrive and do their work. And what he found was treating neurons with just by themselves, without any glial cells around, they don't respond very well to D2-hydroxyglutarate by themselves. But if you treat them in the presence of those glial cells right here, the activity goes up substantially, almost doubles. The electrical activity almost doubles. This is the first evidence that there's ever been that actually the D2-hydroxyglutarate can act on normal astrocytes, normal glial cells, not just the neurons, not just the tumor cells. So this, as far as I'm aware, we're one of the only experimental labs in the world that's actually studying this kind of phenomenon. And that's great because this kind of experiment not only is it useful data, but it also opens up a whole range of additional work that we can do to study this. Why do it D2-hydroxyglutarate? Why does it require glial cells to be present in order to irritate neurons? Why can't it do it just directly? Another thing we did was these are called cortical spheroids. These are nifty little inventions. It's actually a three-dimensional ball. It looks like a dot over there on the upper left. It's 1 millimeter in diameter. This is a mixture of neurons and glial cells and everything. It's almost like a miniature brain that we developed in the laboratory. They're just sitting in these little culture dishes, and this is with the help of some collaborators from the Shirley Ryan Ability Lab and UIC. And what we did was we took those cortical spheroids and just sprinkled glioma cells on them, kind of like salt and pepper. And what was interesting, if we followed them over time, the glioma cells would rapidly migrate into those spheroids. And here's a time-lapse video of that. The glioma cells are in green. They're engineered to express fluorescent green protein. And you can see that these glioma cells rapidly like to crawl into those cortical spheroids. That's another side project we're working on now. Why do they want to go into those spheroids so badly? What's the cortical spheroids secreting that pulls them in? What's handy though, from our purposes, irrespective of that mechanism, is we can culture those spheroids on the exact same kind of a multi-electrode plates that we use to show the video I showed you before with those big bursts. So now the spheroids, which have their own electrical activity, they're sitting on electrodes. And we can measure the electrical activity of those miniature brains, so to speak, with and without glioma cells. And what we found was when we cultured these little mini brains in the presence of IDH mutant glioma cells, TB09 and B23, that's what they are on the right-hand side. Over time, they triggered a lot more electrical activity in these cortical spheroids, these mini brains, than IDH wild-type gliomas. Even though the IDH wild-type gliomas were faster, they're more proliferative, they're more aggressive, the IDH mutants triggered more neuronal activity. And this was a great model to use because, as it turns out, there's IDH mutant enzyme inhibitors out there that are now in clinical trials. Some people listening in on this may actually know about this, they may even be treated with this. Vorasidenib in particular is one. It's also called AG881. It's being explored now in phase 3 clinical trials, and it's a compound that specifically blocks the IDH mutant enzyme from producing that little D2-hydroxyglutarate product, that little neurotoxin. And what we found was that when you culture these IDH mutant glioma cells with those cortical spheroids by themselves, you get a certain amount of electrical activity. That's the gold line, that's the gold circles. But when you treat those cultures with the AG881, that's the vorasidenib, that's the IDH mutant inhibitor, that wipes out the electrical activity and it brings it back down to what we call baseline levels, because we're just the same kind of electrical activity that the little spheroids would have without any glioma cells present at all. So that's pretty good in vitro evidence that the enzyme inhibitor that's being used in clinical trials could be very, very effective at controlling seizures, specifically in these glioma patients. So then we took it into the mouse model where we engrafted IDH mutant gliomas or IDH wild-type gliomas into the brains of these mice. You can see a little tumor. This is the cross section of the mouse brain. And here is the tumor right here that sticks out. It's that dark blue area. And so we'd engraft these tumors into the mouse brains, fit these tiny little EEG caps onto the mouse brain, that's electroencephalography, and just record their electrical activity. And it was striking. In fact, we actually had John Kurz, who's a pediatric epileptologist at Lurie Children's Hospital. We sent him these EEG recordings without telling him whether it was an IDH wild-type glioma mouse or an IDH mutant glioma mouse. And he was able to tell 100% of the time which was which, just because the IDH wild-type glioma mice, they had, pretty minimal EEG activity. That's the upper line. Whereas the IDH mutant ones on the lower, you can see it yourself. In fact, this is what it looks like in real time. So we're looking for these epileptiform spikes, these really big jumps. And here's an example of an IDH mutant glioma engrafted mouse undergoing a generalized seizure. This is what it looks like on an EEG. And you can see those, you see how it's progressing, how those massive firing is going on. It's going on and on and on. And eventually it's going to resolve itself and burn out. That's the so-called refractory phase of a seizure. This is the first time that, to our knowledge, that this has ever been studied in an experimental setting. And in fact, sure enough, gliomas, when the glioma has the IDH mutation, it triggers a lot more of that seizure activity in mice, just like it does in humans. And just like it did in those spheroid cultures, when we treat these mice with that very same inhibitor, the one that's in clinical trials, vorasidenib, AG881, we see a marked suppression of seizure activity. That's the blue line compared to the gold line. And otherwise, all the conditions are identical. So in conclusion, then, what we found is that the IDH mutation really does have a profound effect on seizures. In other words, our understanding of what the IDH mutation is doing is extending beyond just the tumor itself. There's now lots of data to indicate that the IDH mutation markedly affects the entire brain. And that the patient's, the postoperative seizure risk is going to be determined by 4 key variables: whether the tumor is IDH mutant or not, the tumor genotype, whether or not the tumor is in the frontal lobe, whether or not the extent of resection, surgical resection, as well as whether the patient had a preoperative seizure. Those 4 variables. And finally, IDH mutant enzyme inhibitors may actually improve seizure control substantially, which is really important to me, because if we find out ultimately in this phase 3 clinical trial that the IDH mutant inhibitor, vorasidenib, if it does not meaningfully extend patient survival, it doesn't mean we should just drop this drug altogether and say, it didn't work, it's worthless. Actually, it may still be there may be a very, very good role for this drug in helping control tumor-associated epilepsy in these patients who are at the highest risk. So this is my laboratory. Very grateful for all the help and the funding, both from the NIH, NCI, and the Brain Tumor Institute here. And I don't know if we have questions now. We're just going to go to the next presentation, but thank you very much.
Thanks, Dr. Levitsky. So for those of you who are in the room, feel free to write your questions down and pass them this way. We have had a couple of questions come through the chat if you still have time.
Sure.
The first one is, of the 50% who have had seizures, do you happen to know what proportion that seizure was the presenting symptom that led to their brain tumor?
So all of them. In other words, whenever, and that stands to reason, because somebody can have a progressive headache and ignore it over a long period of time. But if you're walking along the street and everything's fine and good, and all of a sudden you have a seizure that hits out of the blue, that's going to trigger a visit to the doctor or the ER no matter what. So all of those patients who had a seizure at the time of presentation, that's what triggered them to go to seek medical care.
And then let's say you had unlimited funding. How would you spend that for your research?
For this project, I assume in particular, or one of the other dozen projects I'm working on?
Any project.
Well, I would say one of the things I really want to delve into more, we're actually already doing this, is really dive in and study and figure out the mechanism whereby D2-hydroxyglutarate is acting on the glial cells. So like I said, it was a bit of a surprise that the neurons by themselves don't respond to D2-hydroxyglutarate. They need glial cells to be sitting right there. That suggests that the D2HG is irritating the astrocytes, which in turn irritate the neurons. How that happens is not yet clear, but that is one of the things we're investigating next.
So a couple more questions. From a neuropathology perspective, do you happen to know any benefits of Briviac versus Keppra?
I do not. That's probably more of a Stephen McGill or Roger Stoop question, or if Jess Templer were here, she could answer that too, but I do not.
And then the other question was related to diet. So we'll also defer to Dr. Templer and some other physicians for that.
I can tell you that the ketogenic diet does not appear to work. We tried that as an experiment, a side project in our laboratory, and it didn't have any effect on IDH mutant or wild type gliomas.
Cool.
Whatever that's worth.
Great. Thank you very much.
So again, thank you for being here. And today we have the pleasure of introducing Dr. Stephen McGill. He's one of our esteemed neurosurgeons within the Department of Neurosurgery here at Northwestern. His focus is on meningiomas, but also skull-based tumors. And today he's going to be talking about his research, which is on meningiomas. Thank you for being here, Dr. McGill.
Thank you, everyone, for coming and for having me and inviting me to talk for the leadership here. It's an honor to be a part of the MBTI. I think it's a fantastic group. And when the opportunity came to come work here a little over a year ago, I jumped at it because just to be part of a team working with people like Craig, Dr. Stoop, the other neurologists, the other surgeons, it's just a wonderful place to take care of folks. And I hope that we can provide good care for you. Shifting gears a little bit, my work is in meningioma, which isn't talked about a lot of times in brain tumor circles as much as meningioma or as glioma. But I want to talk to you a little bit about some of the basics of meningioma, just to put the lay of the land for a lot of the people in the audience who may be more familiar with glioma, and then talk a little bit about how we take care of them, the epidemiology, and then some of the research horizons and what we're doing, and also how we're incorporating that in the operating room as a surgeon. So meningioma is the most common primary brain tumor. If you look here, this is if you look at all intracranial tumors, these are the meningiomas. They account about 39%. There's over 36,000 diagnosed in the US. They're graded grade 1, 2, and 3, and often considered benign. But if you have a grade 2 meningioma, which is 18%, which works out to about almost 6,500 patients a year, which is about half as many glioblastomas, those tumors come back a lot more rapidly. And they happen in older folks. About 66 years old is the median age. A couple interesting epidemiology things. Unlike glioblastoma, which is up here, that's more common in men if it's on this side of the graph to the right, meningioma is much more common in women, almost 2 to 1 compared to men, both malignant but particularly non-malignant. The very aggressive meningiomas, it's not quite as strong, but it's a tumor much more common in women. It's also more common in blacks, whereas glioblastoma is twice as common in white folks. This is more common in blacks, especially the malignant meningiomas. Risk factors for meningiomas: people like children who had leukemia, who had to have craniospinal radiation, oftentimes 25 years later will develop meningioma. So any kind of radiation to the head. At Michael Reese Hospital, I've seen a few patients who had their tonsils radiated when they were kids, and then now they're adults and they have meningiomas. They were doing that for a while back in the day. Any kind of radiation around the head can do that. It's associated, if you have a personal history of breast or thyroid cancer, you're at a slightly higher risk of getting it. There's some familial syndromes, a bunch of them here with known gene mutations, but they're neurofibromatosis being the most common. But outside of that, most of these are sporadic. Research I did a couple years ago, along with a nurse practitioner, Jennifer Viner, back at UCSF, who noticed that a lot of the women who came in had had fertility treatments. And when we compared women who had had fertility treatments with those who hadn't, they had more meningiomas, and they came on at a younger age. And the other place we've seen more of them is in transgender women. So biological males who are transitioning to be women take a lot of progesterone. And if you look at this is an example of a grade 1 meningioma. If you look, this is progesterone receptor staining. And so meningiomas express a lot of progesterone receptors. So there is some hormonal link between progesterone expression and high dose exposure to progesterones in these tumors. Exactly the mechanism of that, we don't totally know. How do people find out about this? The most common is kind of headache or cognitive changes. These tumors grow very slowly, so they can grow to quite a large size like this one over here, and you may not even know it. It's just growing slowly, and then all of a sudden you're starting to get headaches, or dad's just been depressed all the time, or mom's not acting like herself. She's really confused, it seems like, and then ultimately you get a scan and find it. The other ways they happen is if they're right next to a nerve that does something important. So you could have a very small meningioma, but if it's growing next to your optic nerve, or pictured here is the trigeminal nerve that gives you facial sensation, you could have tons of pain in your face and be like, why is this happening? And instead, it turns out there's actually a little tumor growing on that and compressing that nerve. Another about 10 to 20% come around just incidentally. You fall and hit your head and you get a scan and all of a sudden it's there and you never knew. And that's becoming more and more common as we do more imaging. So I mentioned earlier 3 grades. Most tumors, 60 to 80% are grade 1, 20 to 30% grade 2, and just a couple percent are grade 3. Unlike glioma, where some of the survivals are lower, meningioma patients tend to do well as long as their tumor is controlled, particularly the grade 1 meningioma. If you look at overall survival at 10 years, 80 to 90%, progression-free survival, 75 to 90%. That changes with grade 2, where within 5 to 10 years, within 5 years, actually, half of these tumors will recur, meaning you need additional surgery, additional treatment of some sort. Grade 3s are much more aggressive tumors, but they're so rare, we don't talk about them that much. So what do we do when you find out about a meningioma? You get a diagnosis, you have headaches, they find out you have a tumor here, you get a scan. The first decision is observation or treatment. Because they're slow growing, sometimes they're growing so slow or not at all, we just watch with a scan. But if it's one of those tumors that causes symptoms for you, then we move into the treatment arm. For treatments that we have available are either surgery, what I do on a daily basis, or radiation. This is a picture of a gamma knife machine. We can treat it quite effectively with radiation, but radiation just stops it growing. So if you have a small meningioma that's asymptomatic, we've been watching it, and it's growing, then you could treat it with radiation as opposed to a symptomatic one that's bigger or something like that, where surgery may be more appropriate. And the decision making between observation and treatment is really, are you having symptoms and is the tumor growing? Unfortunately, though, that's the extent of what is approved for meningioma. We don't have a Temodar, we don't have tumor treating fields, we don't have any other options aside from what I do as a surgeon or radiation. And so I mentioned earlier, the outcomes are fairly good. These are survival curves. This is recurrence-free survival for grade 1 at 10 years. 20% come back, but 80% of patients are doing well. It's a little different, where about 50% will be back by 5 to 6 years for atypical meningiomas. And overall survival is generally fairly well, unless you have the more aggressive types. As I mentioned, surgery is an important factor in this. And part of the objective of surgery is to remove all the tumor. Meningiomas grow from the covering of the brain and push on it. So our goal is to take out all of the tumor, any involved bone, that as if it invades the bone, and the covering of the brain. And then we replace that and reconstruct things. You can see in this curve that patients who had gross total resection, many more don't have the tumor come back compared to those that have a subtotal resection. And the same is true for overall survival. If you have gross total resection, much better overall survival than a subtotal resection. Radiotherapy, so if it's a small meningioma or one that's eligible for radiation, also very good control, and 95% at 5 years. And we can see that here. So this is an example that I showed you earlier. This is a young woman, 39 years old, who comes in with new progressive headaches, gets this scan. Options are surgery or observation or treatment. This tumor is kind of invading. You can see it here on the covering of the brain, pushing the brain and invading the bone. And so she elected to have surgery. She had a great surgery. So I took out all the tumor. You can see the part that was invading the bone here. I drilled all that out, so there's just a thin bone. And she does very well. That's nice when you're up on the surface of the brain, but if you're at a deep part of the brain near important arteries and nerves, surgery has its limits. This is an example of tumors near the base of the brain, near the pituitary gland and the optic nerve. And if you look at with electron micrographs, here's the tumor, which is this part labeled T, and these Fs are the fascicles of the nerves. The tumors actually invade within the nerves. So if I took out all of the tumor that's invading some nerves, then I'm going to take out the nerve and you're going to lose that function. So surgery has some of its limits, and that's where often radiation comes in. In more dramatic fashion, this is the artery, the carotid artery here in these sections. And you can see that the tumor actually invades in the wall of the artery. So by understanding the biology of the tumor, as it gets down close to that artery, I'll shave it down really close, but I'm not going to try to take it all out. And then you get into big trouble with arteries bursting and things like that. So there are limits to what we can do surgically. This is an example of a case like that. You can see the tumor here. This is the patient's eye here, a 38-year-old woman who had some double vision because some of the nerves that control vision run through this structure, the cavernous sinus. All this tumor is in here. Her pituitary gland here is normal, but it's right there. So if I try to take that out, I'm going to make her a lot worse. So this is a couple other pictures of the tumor, but it's all inside there. So for her, we treated her with radiation. In classic radiation planning, we would just plan the radiation around this. But one of the things that we're starting to incorporate in practice here that's up and coming and newer is a PET scan. And this PET scan uses a molecule that binds to, called dotatate, that binds to some of the receptors, somatostatin receptors that are on the surface of the tumor. So it specifically lights up where the tumor is. And you can see that this is a recurrent patient who had a recurrent tumor. We'll talk a little bit more about him later. But this is some recurrent tumor here. The pituitary gland also lights up. But this really shows us the extent of that. So we can use it to plan radiation. And in patients like ours here with this tumor, some studies from Europe exploring this actually found that if you guided the radiation with these PET scans, you had much better long-term control because you see where the biological activity of the tumor is. Maybe not just what we can see on the MRI, but where is the extent of the cells. So then you can guide your radiation to actually treat the whole tumor and not have any misses. I'm also incorporating that into what we do in the OR. So this is a gentleman who happened to have one of those more atypical meningiomas. And these are the ones that are real problematic. You think, oh, it's a benign tumor, it's not that bad, but this has come back twice. It happened first when he was 45 years old. He had one surgery, had an infection, then had another surgery later after it recurred again, quite a bit later. We treated him with radiation, and now this tumor keeps growing despite radiation. And it's a real problem because I don't have any medical therapies to offer him. We do the PET and we can actually see, yes, there is recurrent tumor, these dark spots. And then this is that scan I showed you earlier. You can see while the very, very clear tumor here in the middle that we could see on the MRI, there's actually PET lighting up quite a bit out around here, suggesting that tumor cells have gone in there. So if I go in and just take this out, I'm not going to do a whole lot of good for him because we can't get all of the cells and get a true clean margin. Unlike gliomas that invade the brain, these rarely invade the brain or not nearly to the extent that that does. So by using the PET, I did actually quite a bit bigger operation like this, where we took out all of this dura, put in a custom bone implant in the back, but we could plan all that based off the PET, and then we could really get a clean margin. So this is a picture of what I do in the operating room. This is me doing surgery with this. And then this is little wand here is the navigation. So I can point, and up here is I have a PET scan in that navigation. So we can actually use in real time to say, hey, have I gotten past the extent of the disease? Even if the tissue looks normal to me, if I see more activity within it, then I want to keep pushing. I want to plan my resection to go further. So that's kind of the basics of meningioma. It's a surgical disease. Surgery is important for both diagnosis and grading, as well as treating the patient. But safe surgery has its limitations when those tumors invade into arteries or nerves, especially along the skull base. Radiosurgery is effective. And then there's emerging things that we're starting to incorporate into our practice in the operating room and in guiding radiotherapy to try to provide better treatments. But what are we doing for research? What happens when you get a meningioma like this one I showed at the beginning that was a grade 1 tumor, but it keeps coming back? It's very invasive, very aggressive. And we don't really have a chemotherapy that can shrink this down. We don't have any medical treatments. So outside of surgery, we do radiation. So grade 1 meningiomas, the general paradigm is we just watch them after surgery, or if there's a residual, we may radiate it. Grade 3, we always radiate. But these grade 2 meningiomas, especially if you take out all the tumor, you get a gross total resection or GTR. It's really controversial. Well, I thought I got all of it out. Should I radiate the patient? The only way, we don't have anything that tells us how should you do that or not, aside from sort of clinical gestalt. And there's tons of papers saying you should radiate and tons that say you shouldn't. This is a list of some of those papers, some in favor of radiation, others saying no benefit. We looked at 565 meningiomas within our cohort that we will study, and I'll talk about from time to time. And we just lined them up by WHO grade, grade 1 up here. If we radiate or no radiation, no difference. Radiation or no radiation for grade 2, no difference. All these p-values are over 0.8. So the question then became, could we find something that would help us guide that? So we developed a gene expression panel using some of these patients, and we identified genes that, based off sequencing of these tumors' RNA, that predicted response to radiation. We then, what this shows on the right is that there's a correlation between the sequencing of these genes and an assay that's very cheap, about $50 to run, that is a special array that can show us, is this reflect the expression of that gene? When we use this gene panel that we developed, we can predict both which patients will have a low risk of recurrence up here, an intermediate risk, or a high risk of recurrence for both tumor recurrence and overall survival. But and this actually, the prediction power of this is much better. The 0.81 of this gene expression score is much higher and more accurate at predicting recurrence than the World Health Organization grade, which is the current standard. We developed a nomogram like this. So if you come to me, and we don't have it in prime time for the clinic yet, we actually have submitted this to New England Journal, and I'll talk a little bit more about that. But we can put your characteristics up and say, hey, you have this risk of recurrence or not based on your gene expression risk score. But what's most exciting about this, and why when we submitted to New England Journal, they were really excited about seeing this, is we found that it actually can predict who's going to respond to radiation. Because you could see, based on the World Health Organization grade, we don't know who's going to respond. It's not tumor grade. There was no difference. We did DNA methylation profiling. There was no difference with radiation there. But if you look at patients who had the either intermediate or high gene expression risk score, if they didn't get radiation, those tumors tended to come back. These are the grade 2 meningiomas after gross total resection. If you radiate that, they're less likely to come back. But when we then stratified and we took all the meningiomas and we stratified by risk and matched them by their World Health Organization grade, the chromosome changes within the tumor, how much surgery, like were we able to get all the tumor out or not? We could then use this gene expression risk score and identify a group that should never receive radiation, no benefit, the low risk, compared to the high risk. And you can see that the patients who got radiation do much better. They survive a lot longer than those who have their tumors come back that didn't get radiation. So this is the first time we've had a gene panel that will actually say you should get radiation or not. And this could be 6 weeks of radiation that could have side effects. It's costly. We can save patients who have a tumor that looks bad under the microscope, but a low gene expression risk score. And those that have, who could really benefit, but maybe we didn't know it was going to be a bad tumor. It looked just like a grade 1, but they have this risk score. They may actually benefit from radiation. So these were 2 examples of tumors, both big tumors pushing on the brain. I took both of them out completely. The one here on the right was a patient whose risk profile of her tumor was very low. The chromosome changes, those types of things. So we're just watching her post-operatively. Both of them were grade 2. The one on the left that doesn't look as big actually had a lot of aggressive changes molecularly within it. So for him, we chose to give adjuvant radiotherapy. But what do we do for my guy that we did that big resection? We got everything out. He's failed 2 surgeries. Hopefully, by using this molecularly guided surgery with the image guidance, we'll get him a long time of progression free. But as good a surgeon as I am, I can't beat biology. So I expect that at some point down the road, this gentleman's tumor is going to come back. And at that point, we don't have any medical treatments. So in addition to the RNA sequencing I was mentioning that we developed this gene expression panel off of, we've been doing DNA methylation profiling on these tumors. And we started studying that in the lab, trying to understand by understanding the biology of the tumor, we can find weaknesses in it and start to target that. So DNA methylation our DNA is stored in the nucleus in chromosomes. We've all heard of those. If you zoom in on the chromosome, the DNA is actually wrapped around these nucleosomes. And if you unwrap it all the way up over to here, the actual DNA molecule itself gets these modifications called methylation to it. Those determine whether a gene can be expressed or not. So there's variabilities to it, but in general, more methylation suggests the gene is not expressed and less suggests it is expressed. So Dr. Horbinski, who you just heard from, has really pioneered using this and brought this prime time to the MBTI, which is so exciting to work in a place where we actually can just send our stuff for methylation just upstairs, as opposed to most people have to send out and it's a month to get back and you don't really know how to interpret it. And methylation is super powerful because by looking at all those marks on the DNA, it can tell you what type of tumor you have. So this is all types of brain tumors. And if you look based just on their methylation profiles, which ones are similar, they cluster into little islands. So like up here is all the different glioblastomas clustered together. And I think over here, the typing's kind of small. This is another brain tumor. If we look at meningiomas, they're this little island right here. And you see all of these meningiomas clustering together. They're close to hemangiopericytomas. And so this is the result I get. It says, we do DNA methylation profiling. It says, well, you have a meningioma. I was like, well, I knew that. So how is it really adding to our care? So we studied these 565 tumors I mentioned when I was at UCSF, and I'm still ongoing collaborations and doing some work here with this and our own tumors here, which we'll talk a bit about, as well as a collaboration with Hong Kong. And we looked at the profile, and again, there's three groups, one group that does really well, an intermediate group, and a poorer group. We studied the biology and found that the group that does the best is called Merlin intact. It has this 22 chromosome preserved. And this protein that's lost in the higher grade tumors actually is still present. So the tumor cells die. So when they have problems, they can still die. But when that tumor is lost, when that gene is lost, that apoptosis is less present and the tumors may grow faster. The intermediate group that we found with intermediate outcomes had a lot of immune cells within them and actually lymphatics growing from the sinuses into the tumor. And they do a little bit worse. And we're working on additional experiments to characterize this immune infiltrate, understand how it impacts this, whether or not these patients would be candidates for immunotherapy. We're not sure that that's the case because of some of the immune features, but I won't get into all that now. But the worst patients, the patients with the worst outcomes are these ones we call hypermitotic. And we discovered several different mechanisms that all activate the cell cycle, cause cells to divide. And that's ultimately what makes a tumor grow, is when the cells keep dividing. These patients, this is their local freedom from recurrence. So how much does a tumor come back? You can see more than half of them will recur at 5 years and many by 10. And this affects their overall survival compared to the other groups as well. So by studying the mechanisms, we discovered 5 different mechanisms that all converge to activate the cell cycle. And what that tells us is, hey, maybe if we block the cell cycle, we can put a peg in the wheel of the bicycle, even if all of the ways get the tumor to grow. These mechanisms are present in 83% of these most aggressive meningiomas we call hypermitotic. So we then went to try to see if we could use drugs that inhibit the cell cycle. And these drugs that all end in ciclib, abemaciclib being the most potent, are drugs that are approved for breast cancer. So they're already approved. So we started treating cell cultures of meningioma cells. And you can see as we add more drug, the amount of cells that are growing in these colonies decreases, and it controls it. We then used a model here. This is a live cell imaging model of neurospheres, kind of like what Craig showed. But unlike the gliomas, the meningioma cells, when you culture them with cerebral organoids, they form these little tumors around the edge, much like a meningioma does for low-grade ones. The high-grade tumors, like high-grade meningiomas, actually invade into the circle. It's a little hard to see, but the circle of cells here at the end of 36 hours is like this, and the meningioma cells have invaded there, but the grade 1 cells all form these little clusters around the edge. And when we treat that model with abemaciclib, we see a big decrease in these clustering of meningiomas around the edge and the invasion into it. We also used mouse models. So these are mice that we put some meningioma cells in the flank and xenograft. If you don't do anything, the tumor cells start growing. But if you treat with the drug, it slows down a lot. And the same affects their survival. So the mice that don't have the drug die of it about a month, and many of the mice with the drug keep living. We've done it sort of experimentally in a few patients with these really refractory meningiomas where we don't really have anything to offer. This is a person on the graph here is the size of the tumor. So he had a surgery, it drops down, then radiation, then bevacizumab, went blind in the right eye because this tumor is behind the right eye. The radiographs are flipped in medicine. But then it kept growing, and it started growing across over to the left eye here. At this point, he started losing vision. We treated him with gamma knife, just laughed at it. Then we started treating with abemaciclib, and the tumor started shrinking down, and his vision came back. So I think by studying the biology, we can actually identify potential targeted therapies for meningioma or things that may become options in the future for patients that have the bad acting tumors that keep coming back. We have several ongoing clinical trials here for that, actually trying to translate that. We just finished the trial where we were a site enrolling this that was run out of MGH, and we enrolled, I think we were one of the top enrolling sites for patients to get on this drug. So we're really looking forward to seeing if there's a benefit in progression-free survival. They just closed this trial. We also have a trial here of immunotherapy. And then we have a trial of tumor treating fields, which I'll mention briefly. And there's another trial where you use that's done at the Mayo Clinic going on now, where they use that molecule that binds to the meningioma, the dotatate, and they couple it to a radioactive particle. So then you just get radiation to wherever those cells are. It takes it right to the cells as opposed to not. This is the schema of the trial that just closed. But I put this up here because one of the problems is there's heterogeneity in the drivers of the tumors. And so if we don't, one of the things this trial we just finished does is they take a look at the genes that are mutated in the tumor. And if you have loss of NF2 or this CDK4 or 6 or CDKN2, all these things that drive the cell cycle, it pairs you with the right drug. And that's really where we go. We need molecular biomarkers to guide our clinical trials. This is the immunotherapy trial. We'll see what comes from that. This trial is led by Priya Kumthekar, who is one of our neuro-oncologists and is really excellent at treating meningiomas. She designed this along with Dr. Stoop using tumor treating fields, which many people are familiar with glioma that have alternating electric currents through the brain, along with bevacizumab, and we'll see how patients do. And the reason this idea came up is that there was a patient with glioblastoma who also had an incidental meningioma. And that meningioma was there after chemotherapy and radiation. But once they started the tumor treating fields, you can see after 8 weeks and 20 weeks, that tumor started shrinking. And so it raised the possibility, hey, this is a totally different modality. It's not a drug. It's a different mechanism of action. Maybe it'll play a role in meningioma. There's some data, a few patients we've had on this where you give bevacizumab, that takes care of some of the edema, but the tumors actually go away. And even when you stop the bevacizumab, the tumor treating fields hold it in check. So we're doing the trial. It's not proven, but we're excited to think is that, when you go from, we have nothing to offer that's proven medically to something that may offer a lot of promise, it's very exciting. This is that last one I mentioned with the targeted lutetium 177. It's done at Mayo Clinic. I think there's a program in New York also doing the same trial, looking for these recurrent tumors. So in conclusion, meningiomas are not always benign. We're incorporating molecular tools into what we do both in radiation and in the operating room. And then by studying the biology of these tumors and doing the experiments that we're trying to do here and that many support, I think we're getting ideas about how can we target these tumors in the future. And hopefully we'll start to see some actual responses in the clinical trials we're doing. As we move forward, by also studying the biology of these tumors, we'll be able to pair the right patient with the right treatment. And that's the most important thing, because oftentimes clinical trials may say like, hey, it showed no benefit, but that's because half the patients didn't have the right mutation or the right biology of the tumor, even though it was a glioma or it was a meningioma, it's not the right pair. So much of what we're doing is taking that biology and matching it up. So just acknowledge the folks here. It's awesome to be part of this team, our leadership, my collaborators at UCSF and Hong Kong are funding, including the Malnati Tumor Institute, which is funding some of the gene panel research that we're doing. And just want, I think you guys investing, the people who support the MBTI and what we do, this was Mike McDermott, who is one of my mentors, who is very passionate about meningioma. When you see people who are caring for patients, invested in moving things forward, it motivates a lot of others. And so by creating that environment where we can fund projects, where we can support students and trainees, it really can make a difference in exciting progress for research, but also exciting young investigators to start studying everything. So with that, that's the end, and I'd be happy to take any questions.
Thanks, Dr. McDowell. So we got another question actually about diet. Do you have any words related to how diet can potentially impact the brain tumor?
So we don't know for meningioma, there's no evidence that I'm aware of that a certain diet will stop it from growing or decrease it. There's an interesting thing, and we're actually working on a study right now. I just came on my way here from a meeting with our students who are leading this. There's evidence that obesity in men is associated with an increased risk of getting a meningioma. It's not like a one-to-one thing, but when we look at men with meningiomas, they tend to be more obese, and this is what we're finding, than men in their same zip codes. And one of the thoughts is that obesity can increase some of the estrogens that our body has based on aromatase, some of the enzymes there. So there may be some correlation, particularly for men, that it raises the estrogen or progesterone levels and can drive tumor origination. But again, we don't really know how progesterones or estrogens can drive meningioma growth, so those mechanisms are very hand-wavy.
So thank you. The second part of that question was related actually to exercise. So, in theory, if someone was fairly healthy, it could reduce some of the estrogen that's naturally produced in their body.
Particularly for men, there's, as you know, like, as I mentioned, these are more common in women. I don't, so, and progesterones can stimulate them. There's no evidence that things like birth control can, but we don't always, we don't know. Those like progesterone-based therapies. So I don't really have exercise is good for you. You should do it all the time. I think it does reduce risk of cancer being in shape compared to not in shape. So I always, when my patients ask, what should you do? I'm saying, don't smoke, stay in shape, exercise, live a good life, have good social things that are important for our quality of life, but I don't have a direct correlation.
So does that mean if you take birth control, is that like,
There's no association that I've ever seen that taking birth control could put you at risk for getting a meningioma. The fertility treatments are interesting because it's very different. You're taking it's not a direct correlation. It's just in our population, we saw this. And so I now ask patients about it because we saw an association. So association and causation are two very different things. Maybe the reason they couldn't get pregnant has something to do with it. And you're taking fertility. We don't know. So I just have a lot of caution, but there's no studies that I'm aware of that say taking birth control shouldn't. But if I have a patient with a known meningioma, like we often see patients who are pregnant and they didn't know they had a tumor, and then all of a sudden it starts growing a lot when your progesterone goes way up. And when you're trying to induce ovulation to harvest eggs, things like that, or you give often very high doses of estrogens and progesterone. And the same with, and I thought it was really interesting when we saw the same thing play out in the trans population, in men who take a lot of estrogens to change their appearance or their gender. Anyway.
Obesity, did you find a high correlation to insulin resistance?
We didn't look at that. Insulin resistance obviously is associated with metabolic syndrome and obesity. And that again was limited to men who would generally have very low levels of estrogens and progesterones. And so we're working on that project now. And there are some historical studies that also show an association that men who are obese have a slightly higher increased risk of meningioma. This is not like you're going to get a meningioma. It's just compared to the general population, it's a little bit higher.
How much of each grade of meningioma are diagnosed each year, and is that why there's less research in meningioma that's happening?
So it's a great question. Two questions. I'll go back to the beginning. There are 36,000 meningiomas diagnosed every year. Too many clicks. 36,000 a year. So you can see the percentages there, 80, 18, and about 2% are grade 3. The reason that most people that the research in meningioma is really unmet and it's been slow to progress is partly because glioblastomas are so bad. So and meningiomas are often labeled benign. So, oh, it's just a benign tumor, forget about it. But they don't see these ones that are really invasive. They don't see a patient that has to go through 2-3 surgeries. And when you have 6,000, 8,000 patients, it does cause a problem, but patients aren't dying in 2 years like you do with glioblastoma. So the severity of that has caused people to, it has caused the research not to be as exciting. People are like, I want to make a difference. Let's do it. Let's do it where we can really make a difference for the most challenging, the hardest hit things. When you care for these patients and they've gone through 3 surgeries and their eye is not working and there's double vision and they're really hard and there's not much to offer. And then you have patients, I did a lot of work publishing on complications. You can just keep doing surgery, but complications go up and you have skin breakdown and you have infections and hydrocephalus and deformities. And it's very difficult for those patients. So, but because in the brain tumor space, I think, and historically the mantra was just, oh, this is a benign disease. We don't really care about it. And it's been much harder to fund. That's changing a little bit, but even when we write grants, it's still much more difficult. So even though it's very common, that's kind of the challenge.
One last question. So why is my pathology diagnosis different from what my neurosurgeon and neurologist say my tumor's behaving like?
So, and I should, it's interesting. It'd be a good slide to include in the background. And it's my first slide. So this is a World Health Organization grade 1 meningioma. When you think about, oh, you have a benign meningioma, you watch it, you take it out, you're fine. Like the lady I did the surgery on, she's fine, she carries on, no problem. That's a benign grade 1 meningioma. We also see tumors like this that are taken out and then come back and are very invasive and growing fast. And so we all who care for patients with meningiomas have patients who everything looks benign. There's not a lot of dividing cells, but when you watch what happens to the tumor keeps coming back. It's growing fast, it's invading. And so in those situations, even though the label may be grade 1, the behavior of the tumor is different. And that's really where I think the DNA methylation and some of these things are, and trying to dive into biology, is identifying these smaller subsets. And as we learn more and more about it, we're finding subsets of patients to explain why that is. And I think things like our gene expression risk score may help. You could have a grade 1 meningioma with a high gene expression risk score, and that's much higher risk. So it's really improvements in our molecular diagnosis.
Thank you very much.
Thank you.
When is the decision made to use a PET scan over the MRI? I know PET scans are a lot more expensive, and do insurance companies typically cover that cost?
So the question was, when do you choose to do dotatate, and when do insurance companies cover it? The way I'm starting to use it here, and this is really just emerging, is in how to use it, how to interpret it. We use it in cases where there's recurrence, like where you've had prior surgeries and you don't know, is this scar tissue that's lighting up or is it active tumor? In tumors that we're going to treat with radiation, where the extent, it's hard to tell the boundaries. Those are the patients I'm asking it. A straightforward convexity, grade 1 meningioma, I don't think it's necessary at that point, because we're going to go in there and take out the tumor. Or if the tumor's invading into a place where I can't take any further, we know we can go up to that. So doing the PET scan isn't going to guide what I do for you. But in places where it can help us, those are the ones I do. As far as insurance, it's approved for neuroendocrine cancers that express somatostatin receptor. And so sometimes we can get the insurance companies to include that. We have some papers. I usually send the papers, sometimes try to cite in my note, based on the data by blah, 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 to get them to push their hand. But it's not a standard of care, and it's not one of the approved indications for dotatate PET. So it's approved for neuroendocrine cancers in the abdomen that also express somatostatin receptor. So there is some trouble getting it approved from time to time. So, and that's why I kind of put it as sort of, we're pushing the edge. This is by no means standard anywhere. This is, something that we've just started, I think, even doing in the last year or two, a little more since I've been here kind of exploring this, pushing this. Okay, I'm standing between us and the food, so let's get something to eat and chat and then enjoy our company.
So we have in the back of the room our dinner, and we're inviting you all to be taking part in that. It needs to have the unwrap off and the saran wrap taken off. And then we have another speaker, probably about 5:35 or so. So please enjoy dinner.
For those of you who are joining via Zoom, we are going to mute the room so you don't have to hear us eating in the background, but please plan to rejoin us at about 5:35.
stage.
Oh.
Okay, everyone, I hope you enjoyed some of that delicious food. Please go back for seconds, thirds, and fourths if you, we have plenty. We're going on to our last speaker, and this is Dr. Tara Kajaz, who will be talking to us today on radiology's role in the care of the brain tumor patients. He's one of our neuroradiologists that we're lucky to have them, they rotate and they actually sit up in clinic. So many of our providers are able to go in and have a, direct conversation when people come in and talk about their films. So thank you for being part of this today.
Thanks, Mary Ellen, and welcome everybody. Again, my name's Tara Kajaz. I'm one of the neuroradiologists working in the Brain Tumor Institute. And my talk is basically just going to be sort of a brief overview of some of the value that imaging has in the context of the care of brain tumor patients. So let's go ahead here. So, one question that comes up, there's a lot of confusion. Even my own parents sometimes can't get this straight. But what are radiologists, right? So we're physicians, we go to med school, we do radiology residency, and then we subspecialize beyond that. So in my case, I did a neuroradiology fellowship and continued in that arena. And we interpret all kinds of different imaging modalities. So ultrasound, computed tomography, MRI, nuclear medicine, a whole bunch of stuff. But in this context, I'm going to kind of keep things focused on CT and MRI. And basically, our purpose is to try to aid in the diagnosis of disease in the initial stages and to monitor treatment progression. A lot of us, myself included, do procedures as well, but that's a little bit out of the scope of this. So how are we different from radiation oncologists? Those are also physicians, but those are the guys, and you may have heard from some of them today, who treat the tumors with radiation. And then the technologists are the non-physicians who actually acquire the images. So they're the ones that run the scanners, do that kind of thing. So this is just a list to give you an idea of how many of us there are here at Northwestern. There's a lot of neuroradiologists on staff. Most everybody on this list actually rotates through the Brain Tumor Institute. And even if they're not in the clinic itself, they sometimes will read follow-up scans on patients as part of other rotations on the neuroradiology service. So all of us are board certified and have this certificate of added qualification in neuroradiology, which gives us additional expertise with advanced imaging techniques, etc. So in addition to the daily consultations that we do with other members of the multidisciplinary team, our neuroradiologists are active contributors and participants in the weekly neuro-oncology tumor board that we have, where challenging cases are discussed.
Can you raise your voice?
Yeah, I'm sorry. Is it, maybe I just need to get closer to the microphone. Is that better? Yeah? Okay. Sorry about that. So with regard to the modalities that we use in the context of brain tumors specifically, CT and MRI are the workhorse modalities. There are advanced MR imaging techniques, and we'll maybe briefly touch on a couple of these. You may have seen some of these reports or heard about these modalities. When sometimes get asked, what's the difference? Why do CT versus MRI? And there's lots of differences between the two modalities. We'll just kind of touch on a few of them. CT is a much faster acquisition. So for example, if you have somebody that's having trouble holding still because they're having discomfort or their mental status is altered, that sort of thing, CT is sometimes the initial go-to modality because you can get the images quickly before the patient moves a lot and distorts the images. The CT scanner has a larger bore, so it's less of an issue for patients who are claustrophobic. It's relatively quiet. One disadvantage of CT is it uses ionizing radiation, just like radiographs do, X-rays. That's one thing that MR doesn't have. And usually we favor CT for the diagnosis of really acute issues. So if somebody has a very acute change in their status and you're worried about, has this individual had a stroke or have they had a bleed? That's the modality we'll do first. MR is much more time consuming. There's, if you've ever seen an MRI, there's lots of different, what we call pulse sequences. They all look, the images themselves all look different because they're favored or designed to look at different qualities that can help you diagnose specific things. The scanners themselves, the MR scanners, are smaller bore, meaning that the hole in which the patient has to go is much, much closer. So as you may be aware, the claustrophobia can be an issue. They're also very loud, these scanners, so patients will have to wear some ear protection. One big advantage relative to CT is that there's no ionizing radiation, so you don't have to worry about the radiation issue at all. But safety stuff with MR is more related to metallic devices, because you're in a very strong magnetic field when you go into the scanner. Anything metallic that's ferromagnetic, for example, can be a problem and can be dangerous. So you get screened for all that stuff ahead of time. These days, a lot of the implants that patients have for various reasons, including some cardiac pacers, are designed to be MR compatible, so patients can still get scans. In the past, that was not the case. So just a picture of a standard looking modern CT scanner to give you an idea of the size of the, you see there's the bore of the scanner and the gantry where the patient lies. This slides in. And then inside this are the detectors and the emitters that spin around the patient and acquire the images. Any physicists or engineers in the room? No? Good. All right. So that I'll be the expert then. That's I always like to check. So again, as you can see from the little schematic here, you go into the scanner usually head first, and then there's this emitter that shoots out these X-ray beams, which are the red lines you're seeing here, and a detector on the other side. And this whole apparatus spins around the patient. And essentially what you end up with is sort of slices through the patient. And now the modern CT scanners are such that you can take that data and you can reformat it in any plane that you want to. So you can look at it from any sort of angle and assist diagnosis that way. So the X-rays, as they go through the body, and this is the same for when you get plain films, they penetrate the different tissues in the body to different degrees. And without getting into it too much, it's just worth understanding that some things will stop more of the X-ray beam than others. So the bone, for example, stops more of the X-ray beam than, say, fat. And that gives us differences in what this stuff looks like when you reconstruct the images after the fact. So here's a couple of images. For those of you familiar with the terminology, this is an axial image. So this is, you imagine the standard is that the patient is lying on the scanner with his or her feet towards you. So that means that this side where the light is now is the patient's right side, and this would be the left. And that's sort of an accepted standard that we use when we interpret these. This particular image is optimized to look at the bone. So you can nicely see the very dense, bright stuff here is the bone. That stopped more of the X-ray beam than this darker stuff, which is a combination of subcutaneous fat, muscle, that kind of stuff, through which more of the beam can travel. This is another CT image, but in this case, we're looking at it in the coronal plane. So the slices are kind of coming through this way instead of this way. And the other difference here is that we've given an intravenous injection of a contrast agent, which contains iodine, and that makes blood vessels show up as dense here. It can also highlight pathology in certain cases. If you contrast that to the CT scanner with the appearance of the MR scanner that we talked about, the fact that the bore was a lot smaller. And once you go into this thing, if you've never been in one, I can tell you, because I have, even if you're not very claustrophobic, it can get to you. There's a lot of little tricks for that can avoid having to sedate somebody. I remember the first time I went on a scanner, one of the technologists told me, they pulled me out, they put a little towel over my face, and they said, just close your eyes and imagine that you're lying at the beach with a towel on your head. And it worked. It sounds silly, but it actually helped. So, but occasionally, obviously, it's difficult for people because they're very claustrophobic or because they're in a lot of discomfort. And we may have to abbreviate the scans or do different sequences that are faster to kind of get them through the process. So again, no physicists, no engineers, so I'm good to talk about this a little bit. The basic idea on this diagram here, imagine this ring is the bore of the magnet. So the magnet, as it sits there with nobody in it, generates a very, very strong static magnetic field. And this is just showing you the field lines in that case. Not that you need to know this, but it's actually typically a superconducting magnet that's encased in liquid helium and then an outer layer of liquid nitrogen. And what that magnetic field does is it makes all the protons in the patient's body align parallel to it. So these red and yellow lines are the field lines. These are the protons in the body, the arrows. So they're all parallel, even though some of them are pointing this way and some are pointing that way. So that's the baseline before you start to do anything. Then what happens is, and the patient doesn't feel anything, but this is when they'll start to hear the banging sound, the loud noise. There's a device that sends radio frequency, basically like radio waves, into the patient's body. And what that does is it disturbs that baseline status that you had before. It knocks these protons out of alignment with the field. And then what happens is that gets switched off and the protons, the baseline static field will pull these back into the original alignment. When that happens, there's energy that's imparted from the patient's body and picked up by these radio frequency detectors, and that information is used to generate the images. So that's the short version of it. And once we have that information, it can be used to generate.
Jared,
Yeah.
hit stop.
You use the microphone, I'm sorry.
Oh, I'm sorry. It's
Or
I keep
Zoom-
forgetting. It's just a
yeah,
little
our Zoom
far from me.
participants.
Yeah. Okay. All right. So just a brief look at some of the different sequences. So you may have seen these names or heard them, T1 and T2. These are a couple of basic workhorse sequences that we have. These are two images, obviously, through the brain. And you can see how they're different. So, for example, the substance of the brain here on a T2 image, the cortex where all the cells lie is bright and the white matter is dark. That's sort of the inverse relationship that you have on the T1. Another thing you might notice is that the cerebrospinal fluid, which is in your ventricles and bathes the brain, is bright on one and dark on the other. A couple of other workhorse sequences. This FLAIR sequence is fluid attenuated inversion recovery. It's a kind of like a cousin of the T2, in which you take the signal from the CSF, from the cerebrospinal fluid, and suppress it or make it dark, and it helps to highlight some pathology in and around the brain. I mentioned with CT that you can give an intravenous contrast agent to highlight pathology. You can do the same thing with MRI, but it's not iodinated contrast. It's a different agent called gadolinium. And these two images are showing you a T1 image before you give gadolinium and a T1 image afterwards. All the normal sort of vascular structures will show some, now they're bright, what we call enhancement. And that's normal, but you'll also see enhancement in areas where there's an abnormality, like a tumor, where the blood-brain barrier is broken down. I mentioned some of the more advanced imaging modalities that we use in the Brain Tumor Institute. One of them is a variation of MRI called MR perfusion. And that is a technique that we use to actually look at the volume of blood within the lesion. And it can help us, amongst other things, try to distinguish what's viable tumor from what's tumor that's been treated and is necrotic because we're dying because of radiation. So here's just a couple of examples of this. The image on this side of the screen, on the left-hand side of the screen, is a T1 image where you've given gadolinium contrast, and this is your brain tumor here. The enhancing stuff, the bright stuff, is the solid tumor components. The dark stuff in the middle of it is necrotic or dead tissue in the middle. And what the perfusion scan does is it helps us to characterize this a little bit further. So this map that I've shown you here is the blood volume map, which tells us how much blood is in those particular areas of tissue. And I don't have the scale up here so you can see, but red is at the high end of the scale. So anything that's red here has a lot of blood in it. So this is telling you there's a lot of blood in the enhancing tissue here. And there are threshold numbers that we use that help us sometimes try to use that to distinguish, like I said, viable tissue from viable tumor, in this case, from non-viable stuff or radiation necrosis. Another sort of adjunct technique we use is something called MR spectroscopy. It can also be used to try to sort out what's tumor and what's radiation necrosis after somebody's had radiation therapy. And without getting too technical, it identifies the chemical signatures within the tissue. And there are some that are characteristic for for particular things. So here's an example. On the right-hand screen here, this little square here is the area that's being examined. So where you see the arrow, this is the sample in the normal part of the patient's brain that we're looking at. And these peaks here on this graph are different sort of chemical signatures that we see in the normal brain tissue. So don't worry too much about what these substances are, but this is a normal brain spectrum. In the same individual, we now shift our attention to the other side where you can see even visually there's an abnormality. There's this area that has some different signal. And we put our little voxel here where we're trying to examine the tissue right in the middle of that. And you'll notice how different these peaks look, right, compared to the other side. You'll notice, for example, this NAA peak here is much lower compared to this choline peak, as opposed to what it should be in the normal side. So just another technique that we can use. Diffusion tensor imaging, another more recent addition to the arsenal that we have, can provide a graphic representation of white matter tracts. And we've got some sort of pretty images of that here. Just to orient you a little bit, this is again an axial brain image, just like the other brain MRI images I showed you. But what it's doing is it's specifically highlighting just the white matter in the patient's brain. And the color coding here has to do with the direction of those fibers in the brain. So there's a convention for this, but anything that's green is running front to back in this direction. Blue is coming in and out of the plane of the screen, and red is going from side to side. This is the same information, the same data from this, just taken and reformatted in the coronal plane here. So you can see now this blue thing, which is something called the corticospinal tract, runs vertically all the way from the top of the brain all the way down into the brainstem. And then these images are sort of fancier, more reconstructed 3D images that we'll sometimes put together. Functional MRI, another technique that's sometimes used, as the name implies, it brings an additional dimension to this, not just sort of structural abnormalities that we're looking at, but we're trying to sort of get a sense of the actual function of particular areas of brain. Again, probably too much in the way of exposition here about the details of how this works, but basically the patient goes into the scanner. The technologist will sort of administer different tests. They'll ask the patient to do different tasks that are meant to test motor function. So it might be finger movement, that sort of thing, or language tasks. And they do a whole series of these. And that what we get on the back end of that is these maps. So we have a standard brain MRI, and then overlaid upon that, you have the different tasks. Each line here is a task, something that was the patient was asked to do. So for example, this top one here, the patient was asked to move his or her fingers, this one, their lips, this one, their tongue. So these are all motor. All we're doing on these three images is looking at the same information, but from three different angles, right? So axially, like this, coronally, up and down, and sagittally, like this. So it helps us know, in this case, what we've outlined are the parts of the brain, the sensory motor cortex that's involved, so that the surgeons can avoid those areas when they go in and do surgery. Same thing here, another set of fMRI images, but the tasks here are all language tasks. So the only thing to tell you here is that you'll notice there's a lot of stuff on this side of the brain and not much stuff on this side of the brain. The reason is that this individual, their language function in their brain is predominantly on the left side of their brain. And that's another potentially useful bit of information depending on the surgery. So, in the preoperative setting, we might do a lot of this stuff to describe the lesion, to locate it, to see if it's near anything that's that could be potentially injured during surgery, and to aid the surgeons in that regard. So here's a couple of images from a pre-contrast MRI study, flare, and post-contrast here showing you the tumor in the brain. And then the overlaid information from the more advanced imaging, the diffusion tensor imaging, which is showing you what's happening to the white matter here adjacent to it. So there's your tumor. And you can see on the normal side, the blue of the corticospinal tract is where it ought to be. On this side, it's pushed away, it's displaced. So you can tell where, important real estate has been moved by this lesion. And then we can sort of, in this case, we're doing the motor stuff to look at, tongue, lip, and finger activation and sort of see where the sensory motor areas are relative to the tumor. So from a planning perspective, it helps. In the post-op setting, often the way this works is a day or two after the surgery, there'll be a new baseline study that's done. And the idea is to see, what, if anything, was left behind. And then that provides a new baseline going forward. With subsequent serial imaging after that, we watch those areas and see what they do. How do they respond to treatment? And we kind of give our input about it in that respect. So, is tumor progressing? Is the patient developing any treatment-related changes? Are they developing radiation necrosis? And this is just a brief example to show you some of this stuff. So the top set of images here, we have two sequences, a flare and a post-contrast, and this is the immediate post-operative scan. So you can see, I know that because there's swelling outside the head, and I can see a new surgical cavity here where that tumor that we saw in the previous images has been taken out. So that becomes our first sort of baseline data point. And then, a few weeks later, the patient comes back. A lot of the immediate post-operative changes have gotten better. All the swelling is gone. A lot of this bright stuff, which was edema or swelling in the brain around the tumor, has gone away. You can see the cavity itself has kind of opened up because that mass effect from all the edema and everything's gone away. But now, if you look carefully on this post-contrast image, at the edge of the cavity, we've got this area where the arrow is that's enhancing. It's bright, and it's thicker than it was on the immediate scan after the surgery. So that's the sort of thing, not necessarily a bad thing, but it's the sort of thing that we zero in on to try to follow and make sure that it doesn't grow, et cetera, and become something else. So we also, in this particular individual, did a perfusion image, and the black here is where the cavity is because there's no blood in tissue or anything else, so it doesn't show up. And we're looking to see basically whether this enhancing stuff has any increased blood volume in it, and it doesn't. So then we follow up. Oh, I don't know why I made that so long, but okay. And then we follow up a few weeks later, and now there are even more changes. So if we compare this early follow-up scan to this one, and you're looking at carefully, you can see, well, wait a minute, this has changed. Now you've got all this new signal abnormality that's in front of the cavity that wasn't there before. But the thing that we were potentially worried about on the immediate post-op scan here, the enhancement, is gone. So that's good news. So, unlikely that was viable tumor because it went away. And this, more than likely, is not going to be tumor. It's probably just going to be changes related to the actual therapy, the other therapies given to the patient. Part of the reason we suspect that is because when we do the perfusion imaging here, again, we're looking to see if there's any increased blood volume in these areas. And this is black. There's nothing there. You can see the scale here, so it's very low, which is a good sign in terms of saying it's probably not an area of progressive tumor. All right, if I do this next year, I'll slow that, or I'll speed that up. All right, so that's just a quick, or maybe not that quick, overview for everybody. Did anybody have any questions? Yeah.
Do you know if perfusion is covered by insurance?
I do not know the answer to that, honestly. I don't know. Does anybody else in the audience?
It
know?
is
I didn't even hear.
in most instances, MRI perfusion is covered under your insurance. However, it is going to be dependent on your specific policy.
For the perfusion, we have to argue and give some reasoning why, when it is or not.
Yeah.
Is the right reasoning? Yes. It's not part of it.
Which is going to be true for any type of imaging, for any procedure.
Oh, I remember.
the following the same type question. Are we limited how many CT scans we can tolerate or have?
CT scans or MRIs, because it's a different,
I know.
okay,
Therefore, I asked CT because of the X-ray.
I see. Well, the CT question, I will say this, that the technology for CT has become progressively safer and safer with regard to the radiation dose. There's been pretty precipitous drops in the amount of radiation that people get these days. There are honestly other factors to consider. So for example, if we were talking about a kid, right, and that has a chronic condition that the kid's going to live for a long time, and will have to have serial CT scans over a prolonged period of time, then that's the sort of thing where we'd say, okay, well, we got to spread these out, or we have to use another modality like MRI, because part of the issue is the cumulative amount of radiation over time, and also the length of time, the patient's length of life. So, it's not, I wouldn't sort of view it as the same sort of issue if you're 90 years old and you're going to need, 5 CT scans in a row versus you're 5 years old. It's a different context, does that make sense?
Yes. So what about the MRI? That is unlimited?
Yeah, there's really no issues with, especially with a non-contrast MRI, there's really no documented issues. Yeah.
Thank you.
Yes.
So building on that, the question about gadolinium.
Yeah.
So you get that each time. I know I've had a lot of MRIs, and that's a lot of gadolinium.
Yeah.
And I recall hearing several years back that there was some study going on to find potentially a different contrast
Agents,
so that
yeah.
it could potentially have less of that.
Right.
You know anything? On that
Those things are ongoing, and there are contrast agents in development. I mean, the issue with gadolinium is a complicated one. In and of itself, it's a toxic metal, but it's bound to another ligand, another molecule that renders it, safe for administration. The problem is that over time with serial scans, some amount of that stuff deposits in the patient's body. There is controversy about whether or not that's really an issue. We can see it on imaging. So if I see somebody that's, had multiple MRI scans with gadolinium in the brain, at least over time, I can usually guess that that's the case because you can start to see areas of signal change. Now, whether that really has any substantial effect is still a bit controversial. So it depends what the indication is. I would say in the context of somebody who's being treated for a brain tumor, I think the benefit far outweighs the risk. The issue may be more for patients that are coming in for, chronic headaches or something like that, where they probably really don't need to get an agent like gadolinium, but sometimes they do.
Do you think that it would make sense to do contrast like every other scan?
It's a tough to make a sort of one size fits all generalization on that. It depends what the indication for the scan is. If it's something like, you're worried the patient is developing hydrocephalus, that their ventricles are getting big, that sort of thing, then that's an indication where you could make an argument they don't really need gadolinium for that. But if you're sort of bound to this pathway and you're trying to assess for things like whether or not there's any tumor progression, you really, there aren't too many ways to do that well without giving gadolinium. That makes sense.
I guess, so I was pregnant and then I couldn't have contrast for
Right.
those months.
Yeah.
And they thought that like after I had given birth and I would have to go on chemo immediately. And that like proved to not be the case because I got contrast and then it showed up differently than what they thought it was. So.
Yeah, I mean, again, it's very difficult to make a one size fits all for that kind of thing.
Yeah.
So, yes.
You mentioned about the MRI versus the functional MRI and about having the functional MRI prior to surgery, giving the surgeon team more knowledge.
Right.
Wouldn't that be routine then almost to do that functional MRI always before surgery? Or when would you not do that?
It's really,
Why would you not?
Yeah, I mean, it's really at the discretion of the surgeon, and to be honest, some of it depends on where the tumor is, right? So if it's in an area that we suspect it's going to be close to stuff that's sort of high value real estate, so to speak, then yeah, it makes more sense. But in some cases, things are in non-eloquent parts of the brain that are not as sort of sensitive or as critical. And in that case, it won't add very much.
Would a
Is that-
surgeon potentially make the decision, oh, I had functional MRI on this patient already for surgery, previous surgery, so I don't need it this time, even though that tumor was coming back and subsequent surgery was needed?
I mean, again, I think that's a tough question to ask. I think probably maybe Stephen, you can speak to
Just
that.
address it, the functional MRI is really important if you're not sure where language is. So like in a left-hand person where 10% of the time, maybe it's on the right side of the brain and we're trying to decide, do you need to go through surgery awake or could we do this asleep? That can really help. And then if you can avoid the surgery awake, I mean, most of us would rather be asleep. So then it's very useful. If you have a tumor up here in the frontal lobe, far from speech, far from anything else, then you don't really need it because it's just going to tell you where speech is, where the motor function, all the examples you showed so nice. So, but if there's a tumor invading in there, and we're trying to plan, can we do this awake or asleep? Or if we're planning a surgery awake and we want to know, like, where am I going to be looking to map? I could spend all day mapping, or we can really try to say, hey, this is where I think it is. Then sometimes we'll do that preoperative. It's very interesting. There's a couple studies that have looked at how function changes, and we know the brain can
Can you
change.
go to the microphone? Sorry.
Yes.
You're having the same problem I did.
Sorry, we're hybrid. So about the functional MRI, if there's questions of where language is or which side of the brain, it's very helpful. Or where the motor cortex is in different parts. Sometimes the diffusion imaging, the diffusion tensor he showed beautifully, or that can help. And then we were just talking about the second part of the question, which was when does the function move? And one of the people I trained with, Dr. Berger at UCSF, we did a lot of language mapping. And he would take a picture every time of the case when he did an awake case. So if you have a left-sided tumor, we take it out, we're mapping your speech. And you take the tumor out right up to where the speech is or where the motor function is, especially speech, I think more common than motor. But what you would find when the tumor came back, we do another surgery awake. Oftentimes that area that before had speech function, the speech has moved. And you could take out a little bit more. And I think he published that 2 or 3 years ago. So that functional MRI you could repeat, which would give you that idea. But when it comes to surgery, really, there's some people that will do surgery based just off the functional MRI, but most people find that it's safest to map because it gives you an idea, but the bold signal is blood flow. It's not down to what we can take out surgically, and where extended resection makes a big difference. How good that surgery is, what we can do for you. Really, mapping is the most important in there. But it is interesting because we do see things move even in adults.
Why would you do both then, both the mapping and the functional MRI?
Sometimes we do. The functional MRI is a little longer. So if we're going to be mapping anyway, and we know you're going to be awake, why leave you in the MRI for another hour? And so I think a lot of what we do in medicine is trying to really, you asked a similar question, I think, about the dotatate for meningioma. It's will this impact what we do? So if your language is on the left side of your brain, or on the right side of your brain, we can then just go to the left side asleep. So that totally changes what we do for you. If we're going to have to go in and map anyway, because we know we're right near there and we're going to be pushing up towards that, then a functional MRI may not change sort of what we do. And then you had to spend an extra, I don't know how long it is, an hour in the scanner, hour and a half, as opposed to being out doing something fun. So, and the cost. And because, medicine is expensive. So is it really worth that? Mary Ellen.
Yes.
When all these patients are getting their MRIs, is there something that, you would recommend when they fill out their long questionnaire that they, indicate anything that they're experiencing that would help you as the reader that never gets to meet them to really make your reading that much more, better?
You mean in terms of their symptoms?
Yeah, like maybe this person's been having headaches for, the past week, is that something they should put down?
I mean,
or
I
they
would
think
just,
that they're,
yeah, as a general rule, I would say whether it's in the context of brain tumors or anything else that we do as neuroradiologists, more is better. More information we have, often we'll get pretty sparse histories, and sometimes it can change the context in which we view things. It's not, a lot of this stuff isn't, it's a yes or no answer, right? It's often not that clear cut. And a lot of other things change as the context. So the short answer is more is better for us. But to be honest, we have access to all the EMR stuff, all the electronic medical record stuff, and it's practice for us, honestly, to review that to the extent that we can beforehand anyway, even if it's not explicitly stated information in the intake form. So, all right. Well, thank you everybody for yeah, go ahead.
One quick one.
Yeah.
On the gadolinium question.
Yeah.
You mentioned that it's possible if you have an awful lot of them that there could be some buildup. What would you notice if there were, other than, I think you said it would be, could be on the MRIs themselves, but
Yeah.
like functionally, would there be anything you'd know, or if it was, what would be the function?
Yeah, I mean, that's kind of, again, it's an area that I think is still under active research. There really isn't a lot of information that's, clearly showing trends. A few years ago, there was issues more related to kidney function. And that stuff, because they've changed the ligands, the molecules to which the gadolinium are bound, there's less dissociation of those two. So there's less breakup of the gadolinium and less deposition. So those agents are much safer. And this kidney issue that was sort of hot in the news for a while has kind of receded into the background. So aside from that, honestly, there isn't anything. I mean, if there were clear-cut evidence that this was a major problem, then we probably would have abandoned use altogether. But I think discretion is a better part of valor, and that's why we say, in the context of somebody who's getting serial scans for a brain tumor, I think we don't have, our hands are tied, basically. We need the information. It outweighs any risk. In less sort of high stakes scenarios, like I said, somebody comes in with headaches or, even a stroke, honestly, it doesn't necessarily add information in all those cases. And I think we've over time become more judicious about who we'll give contrast agents to. But as somebody mentioned, I can't remember if you're the one who brought it up, there are other non-gadolinium agents in the pipeline. So that probably you'll start seeing not too long in the future. So, okay. Thank you very much for your attention.
All right. Hi, everyone. So for those of you who don't know me, I'm Chris Balmacetta. I'm the licensed clinical social worker for the Malnati Brain Tumor Institute. Let's give our speakers another round of applause to thank them for their great knowledge. So normally we would have support group at this time, but given some of the time constraints that we've run into, I'm going to speak a little bit about some of the support resources that are available here at Northwestern, and then we'll also have our friend Vince Rock from the ABTA share a little bit about some of the resources that his organization has. So for those of you who don't know, we do have a dedicated social worker for the Malnati Brain Tumor Institute. That's me. So I'm here to offer any type of support that might be needed for both patients, family members, and their caregivers as well. And that support can be all sorts of different things. That can be emotional support, that can be financial support, and some of the support can also just be knowledge of how things kind of work. On top of that, we also have our lovely new patient navigator, Eliza Delgado, who checked all of you in if you came in person. She's a wonderful resource to help with understanding appointments, understanding where to find certain things on campus, because as you may have found in your folder, we have a very large campus, and so large that we need a map often to help guide patients throughout throughout the campus to find whether it be where to go for their MRI or where to speak to financial counseling or the specialty pharmacy or radiation oncology or all the different aspects of care related to your brain tumor. My role is partly housed through the Department of Supportive Oncology, which is a team of social workers, psychologists, registered dietitians, health psychologists, tobacco cessation specialists, and fertility preservation coordinators. So the majority of patients through the Malnati Brain Tumor Institute will have access to the support services through supportive oncology because as we know, going through a brain tumor is not just the chemo and radiation and surgery that is given to you to treat that tumor. There's a lot more that can impact you and your loved ones throughout treatment. So we're definitely here to try to help improve your quality of life in more than one aspect other than just through medications. Any questions about any of the resources that I went over today, just now? Yes.
Support group?
That was my next part. So as I mentioned earlier, we typically hold support group during this time, but we do have a support group dedicated to patients of the Malnati Brain Tumor Institute. It's held on the third Monday of every month from 5 to 6:30. Currently, it is still on Zoom. Please send an email to btisupportgroup@nm.org if you'd like to join. It's a great way to meet other patients, meet other family members, and meet friends that you probably didn't want to have in the first place, but they're great friends to have anyway. Because we all know that this is the group that no one ever really wants to be a part of, but we're going to support you along the way regardless. Any questions about support group? All right. So I know I briefly mentioned financial assistance. We do have some financial assistance options available. Financial assistance offered through the organization, Northwestern Medicine as a whole. We do have the Patient and Family Assistance Fund for specific needs. And then I also have a wide array of external organizations who can help in different circumstances. Take, for instance, I have a particular organization that can sometimes help with certain aspects related to costs from clinical trials. I'm not going to go over it in detail because it's all very specific, but just know that there's usually something out there that I can identify to help offset the cost of something. It may not necessarily be the first thing you think of, but it would be something to help offset costs related to your brain tumor. Cool. That was the very quick version of support services. Vince, if we're speaking.
Thanks, Chris. Appreciate it. And really appreciate being here today. My name is Vince Rock. I am a senior program manager with the American Brain Tumor Association. First of all, I just want to say what a pleasure it's been here today and to hear some of the great presentations offered by the clinicians here at Northwestern. The ABTA has had a great relationship with Northwestern for a long time, and we're really grateful to continue that. So I'm going to talk just a little bit about what the ABTA is, who we are, what we do. So I'm going to start off with just a little bit of background. We are a not-for-profit organization. We've been around for almost 50 years. We started in 1973 with two mothers who had daughters, just little kiddos who were diagnosed with brain tumors. And these mothers realized there was not enough research being done for brain tumors. There just wasn't any research being done at that time, practically. So they started fundraising, and they eventually formed what would be known as the American Brain Tumor Association. And funding research has been a big part of our mission for these past 49 years, and we continue to do that. I usually have a fancy slideshow that shows how many dollars we've donated for research. I can't remember the figure off the top of my head, but it's many millions of dollars that we've raised through fundraisers. We do our Chicago 5K every year, marathons and things like that. Also corporate donors as well. So funding brain tumor research is a huge part of our mission. The other half that I work on is our patient services side. And what that is we provide free patient services to anyone who's been affected by a brain tumor or their loved ones. So if you're a patient yourself, a survivor, caregiver, friend, distant relative, doesn't matter. We exist to serve you and support you in the brain tumor journey. I'm going to talk just a little bit about some of the services that we offer. So first of all, we have a lot of great educational resources. You might see on your table around there, I left a bunch of our brochures there. There's our handbook for the newly diagnosed, our caregiver handbook, and what we call our primer, which has a lot of fantastic information about brain tumors. There's also a card about seizures. I didn't know that there was going to be a presentation today about seizures, so serendipitous that we brought those along. We also have brochures. You can see sample copies on our table there about specific tumor types like glioblastoma, meningioma, oligotumors, and a whole bunch of others. Treatment options as well. All of our brochures are written in patient-friendly language. They're reviewed by healthcare providers to make sure they are accurate and up to date. And this is scientifically and medically incredibly accurate. They're also medically reviewed by a professional medical reviewer as well. We also offer free educational webinars, about 9 or 10 of those a year on specialty topics related to brain tumors. So we've done webinars in the past that are about meningiomas or about glioblastoma or about skull-based tumor surgery. And our presenters are almost always physicians, and they stick around and they answer questions afterward, kind of like what we're having tonight, which is really wonderful. We also have conferences throughout the year, patient and family conferences, which have been virtual starting since COVID started. So that's been wonderful because now everyone all over the country and even the world can attend these. And we have our big national conference. We just had it in early September, about a month ago. It's a 2-day-long event of just nothing but wonderful presentations like you heard today. And it's really great because there's experts coming from all over the country talking about all things related to brain tumors. There's breakout groups and support groups and just all kinds of wonderful ways to connect. As you know, it's a small community, brain tumors. It's a rare disease, and it's hard to find people who can connect with you in that way. So that's one of the ways we do that. And speaking of which, I work really closely on our social support services. So for example, I maintain our database of brain tumor support groups throughout the country. Northwestern's group is definitely part of that listing, among other groups as well. Insider hack here, a lot of groups have gone virtual, and many of them don't have a lot of people attending. So you can attend support groups in North Carolina or New York or California, and they would be willing to let you in if you don't mind hanging out with people from another state. I know it's nice to meet with people locally, but you do have other options too. So check that out on our website. I also manage our peer mentor program. The way this program works is we have a variety of volunteer mentors. We have over 200 people who are brain tumor patients, survivors, and caregivers or bereaved. And these people are all volunteers who are happy to speak with anyone who is newer to the brain tumor experience and wants some kind of personalized support. So we get called by people every week who say something like, I'm diagnosed with this tumor. Can you connect me with someone with that tumor type? And I can probably do it, even rare tumors too. We got people with rare tumors, among also the tumors that you're more familiar with as well, like the ones I mentioned. We also have a really great online support community. It's a 24/7 access type situation where you can post a comment and other people will respond to it and you get conversations started. And it's a really wonderful way to meet other people and get great information. I'm trying to think if there's anything else that I'm forgetting without my slideshow here. I think that's about it. Please check out our website, which is abta.org. You're also willing to come say hi to me over at that little island over there. I'm lonely, so come say hi. I'd love to tell you more about what we do. Like I said, all of our services are free to anyone who need them. If you want to get in touch with us, there's a little business card in your materials there with our 800 number and our email address. We're also on social media too. Does anyone have any quick questions before I go back to my table? No? Okay. All right. Please stop and say hi to me. Thanks a lot for asking me to speak.
again, we're going to be concluding. We'd like to thank everyone for being part of this today. I talk loud. This evening, and we have a few minutes and we can talk with each other. But we're going to forego the formal support group session because we kind of ran over because we were having such great discussion and questions with the other speakers.
Yeah, and if anyone has any additional questions that they would like to ask either the speakers or any of the staff that facilitated this event, please feel free to email us at mbti@nm.org, or you can always call our clinic 312-695-4360.
There will be a survey regarding tonight's forum, and you will be emailed that, so please fill it out. It does really help us when we plan the next one. Kind of we take some of the suggestions and other critiques that you make seriously, and we all review it together. So we're always open to additional topics that you may suggest or ways to better hold this forum.
Yeah, we'll take all positive and areas for improvement on your survey. So please send those our way. Any questions before we end the evening? Well, thank you guys so much for coming. We really appreciate everyone's flexibility, given that this is our first hybrid event. There's lots for us to learn and take forward to our next, to future events, regardless for this or not. Thank you again. Hope to see you guys next year.
And don't forget to visit Vince. We have to turn.